Python all over the place.
Okay. Hello. I'm Ivan Rossi, and this talk I will quickly resume more than a decade of life with Python and bioinformatics, and so just to show that tiny startup can live thanks to Python, I would like to say. Otherwise, it would have been impossible to do what we did and do. Okay, so my society is called BioDec. It's close to Bologna, and it has been born in 2002 as a spin-off of the University of Bologna. And we did and do bioinformatics, and mainly trying to build products and services out of research from the university, so engineering and trying to build into product algorithms and experience that come from bioinformatics research at the University of Bologna. And we are specialized in a small subsector of bioinformatics, which is protein sequence analysis, and in particular transmembrane proteins, and our workhorse is machine learning techniques. And Python is really everywhere in our infrastructure, in our activity. I have to spend some slides in order to introduce the problem as well, because of the audience, which I think that does not know what bioinformatics is, and in particular what's protein sequence analysis. So we deal with proteins, and proteins are important for the pharmaceutical research, and the most precious things to study in proteins is the structure, because it is there that you find many people, you can find places where you can build drugs that bind into it, that have pharmaceutical effects and so on. Problem. The protein structure is very difficult to determine experimentally. In fact, yesterday, in the protein data bank, you find 100,000 structure, and while protein sequences are tens of millions. And the growth of these data sets is not linear, it's super linear. And so you have to use computers in order to try to extract information from this kind of data set. And the workhorse, and there are also other problems. Information overflow, because we have very large sets of data in terms of hundreds of thousands, millions of things. And it should be high throughput because of the volume and the time constraints you have to respect. And there are open problems in bioinformatics. In particular, for example, it is still impossible to reliably determine structure from sequence. And that's a long-standing problem that has been tackled for 30, 40 years, but even nowadays it's not solved. The workhorse to extend this kind of knowledge is comparison. Comparison between sequences. If you have a sequence of which you know properties, comparing to another one, if you find regions of that sequence, to a first approximation obviously, which are similar, you can say with certain reliability that even the two sequences have similar properties. So you basically extend knowledge by analogy through comparisons. Comparisons, you can think of it as an extended form of Hamming distance between strings. And so pairwise sequence is it's really since you do comparisons, you can use machine learning tools. If you have known structures, for example, and of which you know the sequence, you know properties of this structure, and you can map them on top of the sequence. From these sets, you can build machine learning tools, which, if properly trained, extract general rules from the ensemble, and when you give a new sequence to the tool, it will make a prediction with a certain degree of reliability. And the tools are standard tools of machine learning, artificial neural networks, the Markov model, support vector machine, everything but the kitchen sink. The last part of the information. Pairwise comparison is not enough. You need multiple sequence alignments because it captures information from the evolution of the sequence. To put it simply, for example, if something is conserved through time in evolution and it's always there, it's probably important. If things change wildly, well, you have to think of them differently. I cannot say it is not important, but they have to treat them differently. Sequence profile is just distillation of the information of multiple sequence alignment, which is basically the composition in terms of the symbol for each position of the sequence alignment is actually the main data we use to do prediction. It's the input for the machine learning tool. So, just a question. I want to recall, why Python in 2002 at the 2.1.x version? Because we were coming from different experience, physicist, chemist, computer scientist, and it was an easy common ground. It was expressive. You could do fast prototyping and be productive. It was maintainable. And so it means that you could read it after months. And I say it is still maintainable, but I want to to recall that because at that time, the absolutely dominant language in bioinformatics was Perl. And my brain could not even parse Perl, for example. And there are batteries included, many tools around that. And retrospectively, we were flipping right, since now Python is one of the dominant language in scientific computing and data science. At the time, it was not clear it was going to be like that. Our main workhorse in terms of machine learning, the hidden Markov model, which is a very powerful tool if your system can be modeled in terms of probabilities. There is a certain grammar to the problem, and you can express the problem in terms of limited sequential dependency, meaning, for example, that the next state depends just from what's happened in the previous state. And it's a tool which is has been born, and it's extremely useful if you do prediction on time series. It has been used a lot in speech recognition and in sequence analysis because, you know, the things you find in the sequence actually influence the next one. It's a representation of things which are actually connected in space, so they influence each other. Together with Python and with our data, we built our own tool in the Markov model. Actually, we built our hidden Markov models in Python, and complicated model. Here the scheme of Python HMM we built, which is not a standard one because it takes matrices into input instead of just vectors. And you run the thing through, it will give you an answer. a probability of how well that sequence fits to your model. So you can use it as a classifier or to map a model on top of a structure if the classifier tells you that that sequence is okay for the model. To build that, it would have been impossible without using tools from the Python community. In particular, BioPython, which is a community-developed set of libraries and tools for bioinformatics. It's actually part of a bigger framework of BioX language for any language, supported at the Open Bioinformatics Foundation. And what provided us? Parsers for formats and application output. And that was absolutely vital because many tools you use come from different groups. There are open source tools, free tools that you can use in your pipeline. But I have to say that the output format, you will not call it stable. And unless you have a community that looks at that kind of tool and helps each other in fixing the problem and keeping the parser up to date, well, it would be just a massacre. And the sequence object, which is an object that abstracts the sequence and its data and metadata. You have methods and attributes of your object that you can map information to, and that's the core object in Python we used to store analysis and to do computation, and then conversion of formats, handling of structure, specialized external application wrappers to call external specialized C code, for example, and call it from Python and get back properly passed out to put into your structure. Interfaces to databases. BioSQL is another object that we used a lot, which is a schema, actually, dealing with sequences and everything that relates to sequences that we use to persist data and to do queries of the results on top of it. That we extended it, obviously, to fit our special need, but we gave back to the community, and that's important for us. Useful tool, not just from bioinformatics, what we used for prototyping is a computational pipeline library called Rufus, where you can use it basically to build pipelines. It's a sort of make on steroids. You write functions, put decorators on top of it, and you end up with a declarative file that you can use as a pipeline. You run it, and then something fails, can happen because of sometimes numerical methods have instabilities or whatever. And you can rerun it, and it will just redo the analysis that are needed, not the one, it does not start as make for compiling. It's really useful, especially for prototyping. Actually, in production, we use something different, which has a similar approach, but does not use files, but database. Because we store data in the database and check if something is finished on the database, not on files. And then you can do pipeline handling and use it for scheduling on compute clusters and stuff. With this tool, we built things like this. Our workflow is pretty simple. Actually, it's embarrassingly parallel. You have a huge amount of sequence, and you generate the sequence profiles for that. As I told you at the beginning, you run all your hidden Markov models on top of that and do your prediction, classify, store, and that's the new data that you generate. Or we do something like that, analyze the surface of the proteins in terms of finding regions of importance, in terms of drug design. Sometimes the small things are very important. We had a problem. Profile building, it's CPU expensive. It takes from 30 seconds to 5 minutes for a standard building of a profile. Problem, you have to repeat it for tens of thousands of times for each genome, because you can find 30,000, 20,000 sequences for each genome. And they are GPU weeks. And major genomes are updated every 3 months. So you have constraints. And we are small, and we didn't have many, and we don't have many resources. So we tried to be smart, observing that sequence space is redundant. So sequences are repeated. And the profiles that we built are built incrementally. And the algorithm builds it is deterministic, given the same sequence and the same database of sequence. We built our own accelerator, which is really simple. Just hash the sequence, version the reference protein database. You, at the beginning, store the profile and throw it in the key value store. And now you have a cache. So when you redo the calculation after 3 months or something new happens, if it is already computed and the database is the same, fine, don't compute it. If it is already there but the database changed, we update it. Otherwise, we do the calculation. Very simple, few tens of lines of Python code, but it saved our back because it cut the CPU requirement by 60%. And so we could compete using actually a third of the resources that you had to use if you do the full calculation. And then Python front ends to build a web interface, but this is much more known. Plone, Web2Py, and Galaxy. I actually built a component for Plone, which is a BioSQL browser, which we released to the community, and we used it to build some commercial solution for clients. And it was something like that you could use to search and browse information on the database and associate it to experiments, so the experimental data. Galaxy. Galaxy, it's important because it's a web framework for reproducible research. It focuses on the possibility to redo the same calculation and show it and contribute it to other people or exchange data. It's a major framework in bioinformatics, and it is written in Python, with which you can get data directly from online resources, integrate data from lab instrumentation, do calculation, map it back on public resources, and it's easy extensible because wrapping external tool most of the time is just about writing an XML file with description about the input and the outputs. And it is a tool like that where you have a bunch of tools and an history of your calculations that you can, or a user can program visually, building the framework just by pointing and clicking on boxes and connecting inputs and outputs from boxes using a visual programming paradigm. Close up. What we learned in our experience, actually something that every data scientist should know, but it's, there is so much junk around in the literature, in the data sets. It's unbelievable. It's unbelievable. So many times we had to go through the data and just find that it was the model were trained on just that data set. They didn't generalize at all. And okay, so use always training, testings, and validation sets. Keep something apart always to compare and never touch it for the training. This set should be disjoint. Otherwise, You're just cheating. In bioinformatics, you have to be below 25% sequence identity. Redundancy is the enemy. You have to cut down data, cure the data. Avoid feature bloat. Use feature selection to reduce, if you want, generalization, and always compare to simpler, nearest neighbor methods, because the good ones are really hard to beat. And if you don't beat the nearest neighbor methods, there is no point in using complicated machine learning tools. Don't use an SVM if the nearest neighbor is as good as it. And there is no free lunch. There is no killer methods. Most of the methods work better in some domains, and most of the time data curation is more important than the method itself. So you have to be humble and honest to your data. Otherwise, you cheat, you don't generalize, you don't build good models. And the community is your friend, so you should care for them and give back.
Okay, thank you very much, Ivan. I enjoyed so much this presentation, I didn't want to interrupt. But we have 3 minutes for the questions.
I have 3 questions.
Oh my God.
One is just for curiosity. Who are your clients for whom you work? I mean, they are universities or some companies?
Most of the time, the pharmaceutical companies or biotechs.
Okay, my second question, how do you manage the manpower in the sense that there are many people who have got a lot of skills, in all the, using all the programming, all these stuff. But when you show them a protein structure or sequence, I mean, it's a nightmare for them, right? So how you are able to pick up people who can understand the data and also have the capability to...
Actually, we are very few, and the people which understood the bioinformatics and biological data came from the university, and then there were friends which were computer scientists. And so basically we divided the work and used Python as a common language to build code, and the computer scientists focused more on algorithms and stuff they know, web interfaces, and while methods and bio stuff, it was more for the people coming from scientific computing.
And my third question is that with your experience in Python, do you think an open source drug discovery kind of stuff is possible with Python?
Well. Open source drug discovery is difficult, but mainly to the... It's about people, I mean. You need to have the right brain to collaborate, and then you can do something up to a certain level. And then after that, regulations and experiments, things like that will kill you, and you will need a lot of money, unless there are some public structure that give you funds. I mean, when you hit the labs, the costs just go to the sky.
No, in a sense that at least we can create a framework. Suppose 5, 6, or 10 scientists, they are working on one particular disease or problem, biological problem. So they are sitting in different parts of the world. But if we
can do, but the problem is funding. Someone has to give you the money, unless you are the universities. I mean, people need to pay the bills, and so someone which is interested, either association of people which have the illness, or governments should fund the effort. Otherwise, it's really difficult.
Okay, thank you again.